Imagine your brain as a high-stakes construction project, one where a single misstep could lead to Parkinson's, hydrocephalus, or even spina bifida. Today, we are putting on our hard hats and diving into the blueprints of the nervous system, neural tube developments. By the end of this, you will never mix up the mesencephalon and myelencephalon again. Promise. Picture this. You're on your neurology rotation, and a patient comes in with a newborn who has a severe neural tube defects, spina bifida. The parents are devastated, asking why this happened. You realize this all ties back to the neural tube development, a core concept for USMLE Step 1. Today we are diving into the neural tube forms, the brain and spinal cord, focusing on the primary vesicles, their derivatives, and key structures like ventricles. This is a high yield for step 1 because they love to test developmental defects and their anatomical signs and origins. Let's break it down step by step so you can confidently tackle those questions and maybe even impress your attending on the rounds. Alright, let's start with the basics. The neural tube kicks things off by forming 3 primary vesicles at the cranial end: the prosencephalon, mesencephalon, and rhombencephalon. Think of these as the building blocks of the brain. To remember them, I've got a mnemonic: PMR, prosencephalon, mesencephalon, rhombencephalon. These vesicles are going to split and transform into the major brain regions, and step 1 will often ask you to trace a structure back to its vesicles origin. Let's see what each one becomes. The prosencephalon or forebrain is a big deal. It splits into 2 key parts: the telencephalon and diencephalon. The telencephalon becomes the cerebral cortex, which is your thinking cap, and it forms the lateral ventricles and part of the third ventricle. The diencephalon, on the other hand, develops into thalamus, hypothalamus, and pineal gland, crucial for your relaying signals, regulating hormones, and managing your sleep-wake cycle. To lock this in, use the mnemonic PRO-TD. Prosencephalon splits into telencephalon and diencephalon. On step 1, they might throw a curveball, like asking what ventricle is associated with cerebral cortex. That's the lateral ventricles, thanks to the telencephalon. Knowing this helps you connect the developmental origin to clinical scenarios like hydrocephalus from ventricular issues. Let's zoom in on the diencephalon. It forms the thalamus, hypothalamus, and pineal gland, and these structures surround the third ventricle. The thalamus is your sensory relay station, the hypothalamus keeps your body in balance, and the pineal gland handles melatonin for sleep. Here's a mnemonic to nail it: THP for third. Thalamus, hypothalamus, pineal gland around the third ventricle. THP for third. Step 1 loves to test this with a question about third ventricle obstruction causing pressure on these structures. Think hypothalamic dysfunction leading to temperature regulation issues. Next up is the mesencephalon, which becomes the midbrain. It doesn't split like the others, it stays as one unit. The midbrain is home to the cerebral aqueduct, a tiny channel that connects the third ventricle to the fourth, keeping cerebrospinal fluid flowing. My mnemonic here is MES equals mid plus aqueduct. Mesencephalon equals midbrain plus cerebral aqueduct. For step 1, remember that a blockage in the cerebral aqueduct can cause non-communicating hydrocephalus, a classic vignette they might throw at you. Now we have got the rhombencephalon or hindbrain, which splits into 2 parts, the mesencephalon and myelencephalon. This is where your brainstem and cerebellum starts to take shape. To remember this split, use the mnemonic rhomb-mm. Rhombocephalons become metencephalon and myelencephalon. The metencephalon will form the pons and cerebellum, while the myelencephalon becomes the medulla oblongata. On step 1, they might ask about the hindbrain defect like Chiari malformation affecting the cerebellum or medulla. So knowing these origin is key. Let's break down what each of these becomes next. Let's dig into the rhombocephalon's derivatives. The metencephalon forms the pons and cerebellum. Think coordination and balance, while the myelencephalon becomes the medulla oblongata, which handles vital functions like heart rate and breathing. The neural canal around these structures turns into the fourth ventricle, which is a critical space for the CSF or cerebrospinal fluid flow. My mnemonic here is meta POC, mylo med fourth. Metencephalons equals pons in cerebellum, myelencephalons equals medulla, and together they form the fourth ventricles. For step 1, if you see a question about cerebellar issue or a fourth ventricle obstruction, you will know it traces back to the metencephalon. This is also a good spot to think about CSF flow disruptions, like in Dandy-Walker syndrome, a classic step 1 topic. What about the rest of the neural tube? The portion below the rhombencephalon extend downwards and become the spinal cord. The remnant of the neural canal here forms the central canal of the spinal cord, a narrow channel for cerebrospinal fluid. My mnemonic is neural tail equals central canal. The neural tube's tail forms a spinal cord central canal. On step 1, they might test this with a spinal cord defect like syringomyelia, where the central canal expands and causes neurological symptoms. Knowing this anatomy helps you nail those questions. Let's talk CSF or cerebrospinal fluid, another step 1 favorite. Ependymal cells line the central canal and ventricles, and in the lateral third and fourth ventricles. They transformed into a choroid plexus. This is where CSF production happens. My mnemonic is ependymal equals everywhere in ventricles. Ependymal cells are everywhere in the ventricular system. On step 1, they might ask about ependymal cells dysfunction like ependomyomas, which are tumors that can block CSF flow. Understanding their role in forming the choroid plexus sets you up for the next slide where we will talk about the CSF production. The choroid plexus is the star of the CSF production. It filters blood to make CSF, which fills the ventricles, flows through the system, and eventually exits the central nervous system to join the venous system as blood plasma. The cerebral cortex surrounds the lateral ventricles, while the thalamus, hypothalamus, and pineal gland surround the third ventricle. My mnemonic is choroid creates CSF. Pretty straightforward, right? For step 1, they might present a case of hydrocephalus due to choroid plexus overproduction or a blockage downstream. Knowing the ventricular anatomy, like the lateral ventricles around the cortex, helps you visualize where the problem might be. This is also a good spot to think about CSF reabsorption issues like in normal pressure hydrocephalus. The neural tube doesn't just grow straight, it bends in the sagittal plane to shape the brain. At the 3-vesicle stage, we have got 2 key flexures, the cervical flexure between the spinal cord and rhombencephalon, and the cephalic flexure between the prosencephalon and mesencephalon. My mnemonic is cervical C, cephalic PM, cervical flexure at the spinal cord, rhombencephalon, cephalic at the prosencephalon, mesencephalon. These bend helps the brain fit into the skull as it develops. On step 1, they might ask about the developmental anomalies tied to these flexures, like how a failure in bending could affect brainstem positioning. Think congenital malformations. As the neural tube keeps developing, a third bend appears, the pontine flexure between the myelencephalon and metencephalon. This helps shape the pons and cerebellum's position. My mnemonic is pontine equals post-MM. Pontine flexure comes after metencephalon and myelencephalon forms. Let's wrap up. For step 1, you need to know the vesicles, their derivatives, the ventricles they form, and these flexures. Use the mnemonic we have covered, like PMR for the primary vesicles and THP for third to lock it all in. Next time you see a question about cerebellar defect or a ventricle issue, you will trace it back to the origins like a pro. If you loved this breakdown, hit that like button and subscribe for more USMLE Step 1 tips to help you crush the exam. Let's keep rocking it together.